Rarity Bioscience, where sensitivity matters. To begin SuperRCA analysis, cells must first be broken down, allowing the DNA to be released and isolated. Once isolated, selected DNA regions are amplified through a targeted enrichment process. This ensures that even the smallest traces of genetic material can be reliably detected downstream. One strand of the amplified DNA is converted into a circular template using a ligation probe. A small gap in the new template is sealed using a ligation enzyme. This circularized DNA becomes the template for rolling circle amplification. Rolling circle amplification, or RCA, uses a polymerase to transform the sequence into long, repeated chains, dramatically improving the specificity. To perform allele distinction, genotyping padlock probes are added, each probe being specific to distinguish between wild-type and mutated sequences. Genotyping each RCA loop achieves multi-factor genotyping, meaning that any signal from a misligated probe is suppressed by a majority voting mechanism. During the second amplification, the SuperRCA step, each loop is amplified further based on the genotype sequences, enabling the high sensitivity of the assay. Fluorescent labels are then introduced, binding to their respective targets. The result? A DNA-based particle of micron scale, which is highly structured and highly specific. The target sequence has been genotyped countless times, and the abundance of fluorophores enable the visual distinction of the SuperRCA particles. In the final step, hundreds of thousands of these particles pass through a flow cytometer. Each SuperRCA particle is scanned by a laser and emits light at a specific wavelength, depending on its genotyped identity. The recorded dot plot is a representation of the variant allele frequency, providing the results of the assay in a rapid and quantitative format. The outcome: a high throughput and highly sensitive assay. Rarity Bioscience, enabling the detection of the undetectable.